Hello guys. So today we are going to talk about the this hello guys. So today we are going to talk about the this hello guys. Okay. So let's start talking about I'm sorry. Okay. So today we are going to start the gram-negative bacteria. So Diplococcus, there are four types of gram-negative bacteria. So Diplococci, Cucubacilli, Bacilli, and the comma shape. Okay. So in Diplococci, we have the aerobic. Okay, and uh, if the bacteria is aerobic means it can use the oxygen. Okay. So Diplococci. Okay. Diplococci means it is a double uh, double spherical. Okay. Then aerobic and if the and if the bacteria can utilize the maltose, then it is called the Neisseria meningitidis. Okay. And if it doesn't, then it is called the Neisseria gonorrhea, and Moraxella. Okay. So today we are going to talk about the Neisseria gonorrhea. Okay. So let's start. Where is my where is the Neisseria meningitidis? No results. Okay. So today we are going to talk about the Neisseria gonorrhea, which is a diplococci. Okay, diplococci, aerobic, and non-maltose fermenter. Okay, so yeah, so let's study. So Neisseria gonorrhea. One second. Uh, so Neisseria gonorrhea. Gonococci oxidize only glucose and differ so Neisseria gonorrhea. Gonococci oxidize only glucose and differ so Neisseria gonorrhea. Guys, can you hear me? Pranay, can you hear me? Hello? Hi, can you hear me? Am I audible to you? Am I audible? सुनाई दे रही है मेरी आवाज? I guess I'm audible. So let's resume. Okay. So Neisseria gonorrhea. So gonorrhea oxidize only glucose and differ antigenically from other Neisseria. Gonorrhea usually produce smaller colonies than those of the other Neisseria. Gonorrhea that require arginine, hypoxanthine, and uracil, okay, tend to grow most slowly on primary culture. Okay. Gonorrhea isolated from the clinical specimens or maintained by selective subculture have typical small colonies containing pelated bacteria. Okay. On non-selective subculture, larger colonies containing non-pelated gonorrhea are also found. Opaque and transparent variants of both the small and large colony types also occur. The opaque colonies are associated with the presence of surface exposed on and protein OPA. Okay, so antigenic structure Neisseria gonorrhea is antigenically heterogeneous and capable of changing its surface structure in vitro and presumably in vivo to avoid host defenses. Okay. Surface structure include the following. Okay, this is not important. So pili it has pili, it has also the pore and apoproteins, DMP and lipooligosaccharide. Okay. So, it has a okay. So it has a pili fimbriae. Let's highlight this. Hair-like appendages that extend up to several micrometers from the gonococcal surface. They enhance attachment to host cells and resistance to phagocytosis. They are made of a stacked pinene protein molecular weight. Okay. The amino terminal of the pidgin molecule, which contains a high percentage of hydrophobic amino acids, is conserved. So, the amino terminal of the pidgin molecule, which contains a high percentage of hydrophobic amino acid, is conserved. The amino acid sequence near the mid portion of the molecule is also conserved. This portion of the molecule serves in attachment to host cells. Okay, this is important. So, moisture management process and is less prominent in immune response. Okay. The amino acid sequence near the carboxyl terminal is highly variable. This portion of the molecule is most prominent in the immune response. The pillins of almost all strains of Neisseria gonorrhea are antigenically different, and a single strain can make many antigenically distinct form of pillin. Okay, so por. Okay, so or protein extend through the gonodal cell membrane. It occurs in trimers from pores in the surface through okay see, through which some nutrients enter the cell. Pork proteins may impact intercellular killing of. Okay, this is important. So they may impact the within neutrophils by preventing the phagolysosome fusion. In addition, variable resistance of gonococci to killing by normal human serum depends on whether pork proteins selectively bind to complete to complement components C3b and C4b. Okay, so you guys might be wondering about the. So you guys might be wondering what is the complement proteins. So complement proteins are the proteins which are present in the plasma in an inactive form. Okay, in inactive form. Okay. So when they are activated, they cause the bacteria to to kill to kill the bacteria and yeah. Okay. So C3b and C4b are the proteins. Okay. The molecular weight of pork varies from 32 to 36 kilodalton. Each strain of gonococcus is only one of two types of pork, but pork for different strains is antigenically different. Serology typing of pork by agglutination reaction has distributed 18 syllables of por A and 28 syllables of por B. Serotyping is done only in reference to in reference laboratories. Okay. OPA proteins. OPA. These proteins function in adhesion of glucose within colonies and in attachment of glucose to host cell receptors such as heparin related compounds and CD66 or carcino carcinoembryogenic antigen-related cell adhesion molecules. One portion of the OPA molecule is in gonococcal outer membrane, and the rest is exposed on the surface. The molecular weight of OPA ranges from 20 to 20 kilodalton. A strain of glucose can express no one, two, or occasionally three types of OPA, but each strain has 11 to or 12 genes for different OPAs. Okay, RMP protein 3. So, this protein molecular weight 30 to 31 kilodalton is antigenically conserved in all glucose. It is a reduction modifiably protein RMP and changes in its apparent molecular weight when in a reduced state. It associates with the pore in the formation of pores in the cell surface. Lipo-oligosaccharide. In contrast to the enteric gram-negative rods, see chapter 2 and 15, gonococcal lipopolysaccharide does not have long. Okay, so gonococcal lipopolysaccharide does not have long O side antigen side chain and is called the lipo-oligosaccharide. Okay, so its molecular weight is 3 to 7 kilodalton. Gonococci can express more than one antigenically different lipo-oligosaccharide chains simultaneously. Toxicity in the gonococcal infection is largely attributable to the endotoxic effects of LOS. LOS means lipo-oligosaccharide. Specifically in the fallopian tube, explant model LOS causes ciliary loss and mucosal cell death in the form of a molecular mimicry. Gonococci make LOS molecules that structurally resemble human cell membrane glycosphingolipids. Okay, so the gonococcal LOS and the human glycosphingolipid of the of the same structural class react with the same monoclonal antibody, indicating the molecular mimicry. The presence on the gonal surface of the same surface structures as human cells help gonococa evade immune recognition. Okay, so this gonococcal LOS and the human glycosphingolipid, they are very similar to each other, and yeah, so our immune system identify uh, LOS and the human, and it can, the bacteria can evade the immune system. Okay, so the terminal glycose of the human glycose sphingolipid is often conjugated with a sialic acid. Sialic acid is a 9 carbon 5 N acetylated keto ketolosonic acid, also called as a N-acetyl uraminic acid. Nana. Nana. Oh, you know what, guys? We have a teacher called Nana. Nana, this is the so. Gonococcus do not make sialic acid, but do make cell transferase that function to take Nana from the human nucleated sugar, cytidine 5 dash monophospho and acetyl neuramic acid CMP Nana, and makes the Nana on the terminal galactose of a gonococcal acceptor LOS. This chelation affects the pathogenesis of gonococcal infection. It makes the resistant to killing by human antibody complement system, and interferes with the gonococcal binding of the to receptor on phagocytic cells. Neisseria meningitis and Haemophilus influenzae make many but not all of the same LOS structures as Neisseria gonorrhea. Okay, so the biology of LOS for the three species and for some of the non-pathogenic Neisseria species is similar. Four of the four of the four of the various serogroups of Neisseria meningitis make different sialic acid capsule, indicating that they also have biosynthetic pathways different from those of Gunakuga. These four serogroups chelate their LOS using sialic acid from their endogenous pools. So, other proteins, several antigenically constant proteins of Gonococcus have poorly defined roles in pathogenesis. Lip acid is a surface exposed protein that is heat modifiable like OPA. Okay, lip, this is important. So, okay, so other proteins, several several antigenically contrast constant proteins of Gonococcus have poorly defined roles in pathogenesis. LipH8 is a surface exposed protein that is heat modifiable like OPA. The FBP, ferric binding protein similar in molecular weight to POR, is expressed when available iron is supply is limited, such as in human infection. Gonococcus elaborate as IgA1 protease that splits and inactivates IgA1, a major mucosal immunoglobulin of humans. Meningococci, Haemophilus influenzae, and Streptococcus pneumoniae elaborate similar IgA1 proteases. Okay. Okay. So, so this is important. So, um, genetics and antigenic heterogeneity. Gonorrhea have evolved mechanism for frequently switching from one antigenic form, pillin, OPA, or LPS, to another antigenic forms of the same molecule. This switching takes place in one in every 10 to the power 2.5 to 10 to the power 3 gonorrhea, and extremely rapid rate of change for bacteria, because pillin, OPA, and LPS are LOS actually. This is not epic, this is LOS. This is lipooligosaccharide. This is not lipopolysaccharide. Okay. So LOS are surface antigen surface exposed antigen on gonorrhea. They are important in the immune response to infection. The molecules rapid switching from one antigenic form to another helps the gonorrhea elude the host immune system. Okay. So, the switching mechanism for the pill, which has been the most thoroughly studied, is different from mechanism for the OPA. Okay. Gonorrhea have multiple. They have multiple. Gonorrhea have multiple genes that code for pillin, but only one gene is inserted into the expression site. Gonorrhea can remove all or part of the pillin gene and replace it with all or part of another pillin gene. This mechanism allows gonorrhea to express many antigenically different pillin molecules over time. The switching mechanism of OPA involves, at least in part, the addition or removal from the DNA of one or more of the pentaveric coding repeats preceding the sequence that codes for the structural OPA gene. So the switching mechanism for the LPS is unknown. The antigen and heterogeneity of types are shown in table. Gonorrhea contains several plasmid. 95% of strains have small 50 plasmid. What is plasmid? It's an extra amount of DNA. Okay. So pathogenesis, pathology, and field findings. So pathogenesis, pathology, and field findings. Gonorrhea exhibits several morphologic types of colonies, but only pileated bacteria appear to be virulent. Okay, this is important. Only pileated bacteria appear to be virulent. Okay. The overprotein expression varies depending on the type of infection. Okay. हाँ जाओ उसी हाँ लाइव चल रही है लेकिन करना नहीं है हाँ guys I will be back in short time. Okay. Bye for now.